It's my great honour and pleasure to introduce Professor Elizabeth Thompson, who will give the first talk, title up there. Elizabeth did her undergraduate and her PhD at Cambridge University, did a postdoc at Stanford, then went back to Cambridge as a lecturer in the Department of Pure Mathematics and Mathematical Statistics for about 10 years, and then moved to the University of Washington in Seattle, where she has been ever since. Her area of expertise is pretty much the title there. She's stuck very closely to the same broad area of inheritance, pedigree analysis, populations. Pedigrees and populations, of course, have kind of morphed together over the last 40 years. And Elizabeth has been at the forefront of this sort of work for the last 40 years. She's, without any doubt, the leading expert worldwide on things like linkage analysis and this very topic here. She's had many honours. Perhaps the most important is a fellowship with the National Academy of Sciences, the US, and received many prizes, which I'm not going to mention. Just a pleasure to welcome Elizabeth to speak on this topic.
Thank you for that introduction, and it's a pleasure to be here, and it's a pleasure and an honor to be here to celebrate the WeHi Centenary Symposium. And I am not a biologist, but it's been fascinating the last day and a half to listen to so much exciting biology and new past and current biology. The genomes that I'm going to talk about are much more abstract. They're much more mathematical genomes, but I actually also find them exciting and interesting. So, but Dobzhansky said back in 1973, the famous quote, "Nothing in biology makes sense except in the light of evolution." And a genomics version, a more recent version of that statement might be, "Nothing in genomics makes sense except in the light of co-ancestry, the shared ancestry, the shared DNA that we receive from our ancestors." Maybe that's a slightly strong statement, but at least I hope to convince you that co-ancestry is really important in understanding the genetic data we see today. Some of this work is fairly new technology, and the technical parts of this work are due to a graduate student, Chris Glazner, who graduated last year. And the example that I'm going to show at the end came about because of the comments of an anonymous referee on our paper, which were highly annoying at the time, but actually led to our most compelling example of the success of our methodology. So in retrospect, we're grateful. It's not moving. That might help. Yes, okay. Okay, so the allelic associations, the associations between alleles that we see at different loci that are beloved of the people who do genome-wide association studies, are, of course, the result of co-ancestry. The ones that they're interested in come about because some causal mutation, arises on some genetic background, and because of genetic linkage, remains in association with that genetic background over many generations. And it's leading to an association of the trait that whatever the GWAS person is interested in with that genetic background. But the other main cause of allelic associations that we see in populations, which are the ones that the GWAS people hate, are also the results of co-ancestry. Simply the result of the fact of population structure, that people within population subdivisions are more related than people in different population subdivisions, and that population subdivisions diverge just because of things like random genetic drift. And so here's a very schematic picture. These are all very schematic pictures. Two subdivisions, one in which the blue alleles have high frequency, so tending at different loci to lead to genomes that carry blue alleles in both loci. And the other one happens to be a population where the red alleles have high frequency, so that the red haplotypes or genomes occur in higher frequency, so that in the mixture we see an association of blue with blue and red with red. And in both cases, whether it's the one that you want in trying to map a gene or the one that is just population structure, they are both forms of relatedness. They come about because of the relatedness, the co-ancestry of the DNA. And all the phenotypic similarities that are genetically mediated that we see between individuals are again the result of shared ancestry of DNA. Going back to Mendels First Law, we know that the genome descends generation to generation, and that if we have a fairly recent common ancestor, for example, me and two of my cousins at this particular locus, for example, suppose we received identity by descent DNA that descended from grandfather to his two daughters to me and my cousin at this particular locus, then with very, very high probability in this case, because it's only a very few generations, that DNA is going to be of the same allelic type and lead to phenotypic similarities for things that are controlled by, affected by this particular locus. A lot of people are worried about gene identity by descent. What do we mean by a common ancestor? Because, of course, if we go far enough back, all our DNA traces back to some common ancestor. I think just the important thing to realize is that when we're talking about similarities among individuals, we're talking about it relative to the population at large. And there is no absolute measure of identity by descent. It just is more recent identity by descent than is present at random in the population, leading to similarities among relatives over and above that you see in the population at large. And I'm not going to try and be more definitive than that. But we don't need to have specified a pedigree in order to have identity by descent. We are all related. The relationships among individuals are out there, and the patterns of similarity that we see among individuals in our population results from their co-ancestry. We used to worry a lot about pedigrees. Terry mentioned pedigrees. But the fact is that pedigrees are really a very weak prior on things. This is a figure from a fairly recent paper by Bruce Weir and Bill Hill, when they were just plotting the proportion of genome shared, human genome shared, among individuals of specific pedigree-type relationships. And for example, just to take an example, maternal first cousins, like I had on that previous slide, on average share 1/4 of their maternal genome identical by descent from their grandparents. But 2 or 3% of first cousins will share less than 1/6 of their genome identical by descent, and another 2 or 3% will share more than 1/3 of their genome identical by descent. There is a very broad variation. And furthermore, pedigree is a very weak thing in terms of telling you what that proportion is going to be, or conversely, that proportion telling you what the pedigree relation is going to be. There's a huge overlap, even here between my first cousins and my first cousins once removed. Just telling me that the proportion is somewhere in the range of 20% gives me no evidence as to which of those two relationships it should be. Okay. On the other hand, pedigrees have another bad thing. They're really too strong. They tell me too much if I insist on using the pedigree relationship. You've all done some basic genetics and know that with the probability you share identical by descent with the first cousin, and the locus is 1/4, and the probability you share with your first cousins once removed, for example, your cousin's child is 1/8. But if I do everything in the context of the pedigree, I cannot share with my cousin's child without sharing with the cousin. Given I share with my cousin, there's a 50% chance that I share with my cousin's kid. Whereas if I don't share with my cousin, there's no chance within the context of the pedigree that I share with the cousin's kid. So there are a lot of joint constraints which really don't work when we go to the population level. Because at the population level, our pedigrees that we ascertain for a particular disease or traits are very often biased. They're definitely incomplete. And there could be a lot of other relationships going on in this pedigree. That would mean that I could share with my cousin's child, Dee's child, for example, maybe through his spouse, without sharing with my cousin. So pedigree relationships are too weak in one sense and too strong in another sense. Just as an aside, you really can't validate pedigree relationships. A lot of people say, well, now we've got genome data, we'll just validate the pedigree relationships. Beyond the range of SIBs and full SIBs, parents, offspring, where you can, of course, you really can't do that, again, because of that variance that we saw on the previous slide. So before we had genome data, we didn't have anything else other than the pedigrees, so we had to worry about the pedigrees. But the basic message that I'm going to try and tell you today, that now with genomic data, we can infer at specific locations in the genome, which are the individuals who jointly share that segments of genome identical by descent, And once we know that, the pedigree is not relevant anymore. Sorry, I keep turning back there, but I got it all here. Okay. Okay. Once I know the identity by descent, I can compute any sorts of probabilities of data that I might want to compute. So here again is my little example, my little schematic example of my cousins, and I have data, for example, on these 3 individuals here. And all that matters is that I share this red one, identical by descent, with my cousin C, who shares this pink one with his brother D, and that's all I need to know. In the sense that if I now wanted to write down the probability of these 3, for example, SNP genotypes, C is little a, and me and D and I are A, B, well, what can we see? We can see the red one's got to be little a, the pink one's got to be little a, the other 2 have got to be little b. I've got 2 A's and 2 B's, so the probability of A squared times the probability of B squared, and that's all there is to it. If somebody then comes along and tells me, well, actually, I know there were some other relationships here, so that actually this pink one is identical by descent to this red one from some really recent common ancestry, then all that does is collapse the red and the pink one together. And C now has 2 copies of the red one, and I share that red one with him, and as does D. And so now C is AA, the red one's better be A, and the other 2 had better be B, and now I just have 1 A and 2 B's. And if I have some population model or some population data that enabled me to tell which of these, tell me something about the identity by descent of the pink and the red one, some probability that says it is or it isn't identical by descent, then I can incorporate that information too and get probabilities in my data. Those of you who've met this sort of thing will understand this equation, and other people don't need to. The point is that I can also just write down the probability if I have a model for a trait, a quantitative trait measurement that I might be interested in, like cholesterol level or blood pressure or whatever it happens to be. That is a function of the types of the allelic types that are carried at this locus, then I can equally compute the probability given the pattern of identity by descent, given, if you like, these red and pink and blue bullets, without ever going back to the pedigree at all. So an identity by descent model provides the probabilities for the SNP marker data, and conversely, if you give me SNP marker data, then I can make inferences about the IBD, and I can do my genetic analysis without ever looking at a pedigree. And with modern genomic data, identity by descent can be inferred from genome-wide data pretty well with the more and more SNPs, particularly in humans now, and less easily in other organisms. And basically, this is because the segments of identity by descent are very large when you think about things on a base pair scale. This goes back to the work of my very first PhD student, Kevin Donnelly, not the more famous Donnelly, but Kevin Donnelly, back in 1983, who way ahead of his time was thinking about some of these problems, I think. We all know that if I think about a single locus, Mendelian segregations has a factor of a half every generation, half, a half, a half, a half, they soon pile up. And by the time I get to these red individuals, the probability that they share at any particular locus is only 5 in 10,000. And by the time I get down to the blue individuals, it's only 2 in a million. But we have an awful lot of genomes. Surely we're going to share somewhere. Well, no, we're not. No, quite likely we're not, because the segments that we do share are large and few and far between. And that's because if I am in a state of identity by descent, these red meioses have all lined up so that I get the same genome from some ancestor here down to the red guys. The probability that identity by descent is going to break whenever any one of those M meioses, say there are M meioses there separating them, recombines. It's going to break the IBD. So that over M meioses, it's going to go down as M times as fast, and the segments of identity by descent are going to go down as 1 over M, as opposed to the identity by descent at a particular locus, which goes down exponentially. And this means one can do various mathematics and get that for these red guys, the probability they share something in their genome of 30 times 10 to the 8th base pairs, 3 times 10 to the 9th base pairs, is about 15%, still quite high. By the time you get around to these blue guys, it's only 1 in 1,000. But if they share, if there is a segment, it's of the same order of magnitude and it's millions of base pairs long. because, again, the identity by descent is only broken at a rate of 1 over the number of meioses, not exponentially. So these identity by descent segments we see, even in remote relatives, even separated by tens of generations, are rare, but they're not short. They're millions of base pairs long. In this sort of context, the human genome is really short. And this means that there's been a lot of work, lots of people doing all sorts of algorithms to infer these segments of identity by descent in populations. And an awful lot of these, they look at similar, some of them are model-based, a lot of them just look at similarity of haplotypes. Many of them just did take pairs of chromosomes. They pre-phase their data statistically or genetically and just try and look for segments of identity by descent between haploids. Others do look at the genotypic data on individuals and try and look at 0, 1, or 2 identity by descent between 2 individuals. But I think we want to do more than that. Because when I look at you and us, I would guess that for most of us, our parents are more related to each other than I am to any one of you, or that you are to each other. That is, identity by descent within individuals, between the 2 genomes within individuals, is just as likely as identity by descent between genomes. So if I'm going to worry about all the possibilities of gene identity descent, even between a pair of individuals, the 2 genomes of a pair of individuals, I'm going to have to think about all the 15 possible ways of partitioning the 4 gametes into the ones that are identical, ranging from the one where all 4 are identical to where all 4 are not just the ones where you have 1 identity between or 2 identities between, but also the ones where you have identity within an individual. So, okay, 15 still isn't very many, and let's see what we can do with it. First thing, we're going to have to use the information along a chromosome. We can't just look at this locus SNP, this SNP, this SNP, this SNP. SNPs are not very informative in that way. So again, the key important point is that identity by descent is very slowly varying, again, thinking on a base pair scale, as we go across chromosomes. And here, again, is a schematic small example, 2 individuals, A and B, and a rather simple example where their mothers are related some number of generations ago, their fathers are related some numbers of generations ago, but in this particular picture, their mothers are not related to their fathers. So there's just 4 possibilities. There isn't any identity by descent among the gametes, or they could share their maternal gamete, or they could share their paternal gamete, or they could share both at any particular locus. As I go along a chromosome, suppose they're sharing their maternal IBD, but again, if there's a recombination in any of those meioses, that will break, and I'll lose that IBD. Or, if I'm not sharing, with a much smaller probability, but because now all of them have got to line up, but it could happen that with some probability there's a recombination in the paternal ancestry and they become both IBD. So we can move among the states, in general the 15 states, here only 4 possibilities, according to some probabilities that depend on the probabilities of recombination in the ancestral lineages that come down from the common ancestors to the current individuals. So in general, if I've got n individuals, they have 2n gametes, or haplotypes, if you like. I like to distinguish the actual DNA from the types of the DNA, so I tend to call them gametes, a maternal gamete and a paternal gamete. And at any particular locus, it's just one of those 15 states. So for n individuals, the state of all possible partitions of 2n objects. And as you go along the chromosome, that state is going to change due to recombination, and you're going to move gametes out of being IBD or into being IBD or between elements of the partition in general. So this is a slightly busy slide, but basically it says we can set up a model without, again, don't worry about a pedigree. We can set up a model that will now enable us to infer the identity by descent from our marker data. So we're going to need a model for the process of identity by descent along the chromosomes and a model for the data given the identity by descent. So in previous work, and it's published, I'll give you the references at the end, we've set up a nice model that gives us among 2n exchangeable gametes, so not just for pairs of individuals, but for n individuals, a model that just has one parameter that measures the point-wise, pairwise level of IBD, probability of IBD between gametes. And then we have a model for changes in that identity by descent that maintains that point-wise distribution. And again, I'm not going to go into the details. But we do have to have a scale at the rate of things to which change, because recombinations happen at some rate. You can think of it either in base pairs or centimorgans. Genetic distance is probably easier, but base pairs is what we tend to have. But something that overall scale controls the rate of change of identity by descent along the chromosome. And that'll give us a model for that identity by descent. And then we can have a model. I've already told you really what the model for the data is given the identity by descent. Things that are identical by descent should have, with very high probability, the same allelic type. And if they're not identical by descent, and that is they're not more similar than random things chosen from the population, then we can assume, again, it's something of an approximation to be of, quote, independent types. So if I have I do assume, at least we do assume, that we have good knowledge of our markers, so that we know at least with reasonable accuracy, the allele frequencies of our markers in our population and the marker positions, then we basically have our model. With high probability, you should always allow for some fuzz in your genetic model. So we add one extra parameter, which is, we call it error, but basically it says that things that there may have been a mutation, there may be something wrong, so there might be things that do descend from a common ancestor, but are observed of different allelic type. But some of you, certainly the more quantitative among you, will have met hidden Markov models, probably not in this context, but in other contexts. Hidden Markov models are now everywhere, and that's what we've got here. We've got this underlying hidden process of identity by descent along a chromosome, and at every locus we have data on our individuals, and the information about what happens along the chromosome comes from our model for IBD. The joint information on our SNPs comes not from direct dependence between them, but by dependence through the shared co-ancestry, the identity by descent. So this isn't a pedigree-based model. It isn't it's not a real model in the sense that Mendelian genetics used to be real models, but it gives us a very flexible prior model for the patterns of co-ancestry that we see among gametes. And with our modern genomic data, the data are going to tell us, the data are very powerful, can tell us which are the IBD segments among there. So having a rather flexible prior model is actually good, not bad. And so now we can run it. We have standard programs that do it, and we had a paper that did a lot just looking at pairs of individuals, where it's very easy because you have just 15 states. If you have 3 individuals, you can still run it. There's just 203 states at each locus. That's okay. 4 individuals, well, we have 8 gametes. Now there are 4,000 states, 4,000 possible partitions. It gets to be a little bit problematic, particularly to do chromosome-wide. But that's not going to get us anywhere, right? That's not going to get us anywhere. So we need to be able to deal with large numbers of individuals. We can't do this hidden Markov model for large numbers of individuals. Well, so what do we do? Well, statisticians would all say use MCMC. Several groups of people, two lots of people in my group also, have valiantly tried to use MCMC on this problem. It's not tractable at the moment. What has worked is this idea that's due to Chris Gleisner, known in statistics as product of approximate conditionals, being used in other areas slightly differently, where the idea is that you build things up successively. So we basically add an individual at a time. We have a state of IBD across the chromosome among k-1 individuals, and then we add the kth individual. Okay, and we go on, and we do it lots of times, and we do things in different orders every time. We don't need to go into the details. One important thing is that you've got to get a valid state of IBD. among your n individuals. Think of just three individuals. 15 states between A and B, 15 states between B and C, 15 states between A and C. 15 times 15 times 15 is more than 3,000, it's there. But only 203 of those are valid. Okay, so you can't just blindly go ahead and say, well, glue things together. You have, so when you add your kth individual, and in particular at the stage of adding the identity by descent between the kth individual with some previous individual AI, you have to check the restrictions on your hidden Markov model with all the previous IBD you've got already. But there are two beautiful results due to Chris Glazner. One, that you only need to check trios of individuals, which is amazing. It's very easy once you see it, but it's amazing. You still do have to check all trios of individuals, but that's feasible. And that this above process that he had of restricting the hidden Markov model will always provide a possible trajectory of identity by distinct states across the chromosome. And this was actually, it was in press. It actually came out just last Friday, advanced publication online. The journal issue isn't out quite yet. So we had, and in that paper, we had various examples. The one I'm going to tell you about is a simulated data example, which isn't so interesting in some sense, but it has the advantage that we know the truth, so we know how well we're doing, right? So we simulated descent to 3 groups of fairly closely related individuals, which also had much more remote relationships between the groups. We simulated a quantitative trait at a particular locus, at some particular descent of DNA. Conditional on that descent of DNA at this causal locus, we simulated descent across the chromosome, and then we populated our chromosomes with real haplotypes to make them realistic, to give us SNP marker data on our 31 individuals. And then we ran all our methods. I mean, this is a rather abbreviated version, but we ran our methods. And we said that if we can phase these 31 individuals, which we could because we'd simulated them, then in fact we got the joint 31 individuals IBD state across the entire chromosome, essentially 100% accurate, which was amazing. But in real life, we tend to have unphased data. We didn't do quite so well, but we still did pretty well. And we were pretty pleased with this. But then came the comment of the referee, which says, okay, you've got a quantitative trait. You need to compare this with other methods of quantitative trait mapping using inferred identity by descent. And I don't know if the referee was one of the authors of this paper, but this is the canonical paper that does that, a paper in 2011 by Day Williams et al. Okay, why were we upset about this, apart from the fact it was quite a lot of extra work? The reason why we were sort of slightly upset was that that's a pairwise method. Our whole point had been to get the joint IBD among 31 individuals, and now the referee tells us, well, compare it with a method that only uses pairwise identity by descent in the first place. Because the standard method basically fits these quantitative trait models that use these pairwise identity by descent matrices between pairs of individuals to give you the covariance in quantitative trait data in your individual. Well, but you can still do it. If I have my true IBD, which we did because we simulated it, I can take that true IBD and say, okay, pairwise among my 31, for each pair of my 31 individuals, what identity by descent does that give us? And what pairwise kinship does that give us? They share 0, a quarter probability, 50% probability of handing on the same one, or 1 if they're all identical by descent. So I can compute it. Secondly, from our realizations of joint identity by descent that we have, we can do exactly the same thing. From each realization, we can look at all the pairwise IBD, all the pairwise estimated pairwise kinships. And then, okay, this is our method. We can then use the methods proposed by this paper, which is, I think, the current state of the art for doing local IBD methods, to get their matrix. And as they commented, since the true value should always be 0, a quarter, a half, and 1, you can also take your estimated values and constrain them to be a quarter, a half, 0, and 1. And so we applied that to our example. And this was where we got really happy again. We never wanted to do quantitative trait pairwise mapping, but here's what you get if you get it. if we knew the true IBD, which we did if we simulated, you get the solid line. If you use our method on unphased data, not assuming we know the phase of individuals, we get this dotted line. And in fact, it makes almost any difference whether we constrain it to be 0, quarter, half, or 1 or not, so I didn't put both lines on the map. If we use what I do believe are the best other methods out there for doing this, you get this line. So yes, it's finding the signal, but it finds a signal. This is where the trait locus actually is, where there's a lot of fairly complex IBD, but it nowhere picks up anything like the same patterns. Same pattern, but much less strength. And furthermore, if you do what they advocate and constrain your identity by descent to be 0, a half, quarter, or 1, you get into real problems here in the middle because of the complex patterns of IBD, and essentially you lose the signal completely at the trait locus. So that it could be that there are other pairwise methods that could do better, and my colleague insists I say that, but I very strongly believe that our improvement is due to using model-based inference of the joint IBD and just constraining that joint IBD back to the consistent patterns of pairwise IBD. So here's the summary. We do still want to do descent-based analyses, but really we don't need the pedigrees we used to need in order to do them. We need identity by descent, we need related individuals to understand what's going on, but we don't necessarily need to have the pedigrees, and we may not even want to use the pedigrees if we have them. What we do need is to be able to a good flexible model for identity by descent across the chromosome that allows you to look not just at joint IBD and not just at individual SNPs. Our genomes are not 10 million unordered SNPs. We need models for the descent of chromosome segments and for identity by descent across the genome. And in general, under more complex models, we need jointly consistent identity by descent IBD states. But this was the new thing that came out of the example that the referee insisted we do. Even if you want to use something that's only a pairwise related measure, you're going to do an awful lot better if you use an estimate that's informed by model-based inference of joint IBD. And with inferred identity by descent and the modern genomic data that we have the power of using related individuals, which is really what genetics is all about, or inheritance is all about, or evolution is all about, without the constraints of a pedigree information. And thank you, and there's some references.
Thank you, Elizabeth. Time for a couple of questions. There is one here. John.
I guess the other reason for having pedigree information is that it can also give information about environmental effects as well as genetic effects.
Yeah. A good point, although there may be other ways of getting environmental shared environment that's not necessarily, you could have household information or village information or without necessarily having the pedigree relationships. But yes, the models that I've talked about so far, the models we've looked at so far, really haven't looked at common environment, which is an important component of things.
Second question. Okay. Thank you, Elizabeth. Thank you very much.